Okay, so welcome back to this next video in which we are discussing phospholipase C enzymes. Okay, right. So we're in the process at the moment of discussing phospholipase C beta enzymes and their modes of regulation. Okay, so we've discussed that phospholipase C beta enzymes are actually capable of localizing themselves at the underside of the phospholipid bilayer by themselves. Okay, so they don't need something else to help them bind to the underside of the phospholipid bilayer. They can do that themselves. Okay, and they're continuously at equilibrium between floating in the cytoplasm and being bound to the underside of the plasma membrane. Okay, then what can happen is they can be activated by alpha Q/11 family alpha subunits of heterotrimeric G proteins, which are in the on state. And what these do is they bind to the coiled domain and also the H-alpha-1, H-alpha-2 domain and cause the reorientation of the phospholipase C beta enzyme so that you bring this tin barrel-like domain up to the underside of the phospholipid bilayer, so that it's adjacent and sitting right underneath it. Then what happens is the negative charge on the XY linker gets repelled by the negative charge of the underside of the phospholipid bilayer, and that moves the XY linker out of the catalytic site, so that now that catalytic site is available, and what can happen is a PIP2 molecule in the inner leaflet of the phospholipid bilayer can stick its head in there and get its head chopped off, producing diacylglycerol and IP3. Okay, and then that's how we've activated our phospholipase C beta enzyme. Okay, now, what I also want to talk about is the G-beta gamma subunit can also activate phospholipase C beta enzymes in much the same way as the alpha subunit can. Okay, so G-beta gamma can activate certain phospholipase C beta enzymes. It's not as good at doing it as the alpha Q/11 subunit, so you require larger concentrations of G-beta gamma to produce a significant activation of phospholipase C beta enzymes than you do alpha Q/11 subunits. Okay, but G-beta gamma is capable of activating certain phospholipase C betas. Now, the major one that it's capable of activating is phospholipase C beta 2. Okay, so this is the best one that G-beta gamma is best at activating. Okay, but there are also some that it's slightly less good at activating. So phospholipase C beta 1 and phospholipase C beta 3 can be activated by G-beta gamma, but they're just harder to activate by the G-beta gamma. And again, phospholipase C beta 4 is almost entirely resistant to G-beta gamma. Okay, so it isn't really activated by G-beta gamma. Okay, so you might say, okay, so we're going to activate these GQ/11 heterotrimeric G proteins. The alpha subunit will contribute to the activation of the phospholipase C betas, and the beta gamma subunit will contribute as well. Okay, but the reality is that when you activate the GQ/11 cascade, although you produce lots of alpha Q/11, and that alpha Q/11 is sufficient to activate the phospholipase C beta enzymes, the amount of beta gamma that you produce, which is the same as the amount of alpha Q/11, that's too small an amount of beta gamma to actually have a significant activating effect on phospholipase C beta enzymes. Okay, so you actually have to activate a different cascade to produce a high enough amount of beta gamma complexes to actually get significant activation of phospholipase C beta enzymes. Okay, so you actually have to activate GI/0 heterotrimeric G proteins, i.e. heterotrimeric G proteins where the alpha subunit is either one of the G alpha I subunits or it's G alpha 0. Okay, so let me just explain this. So when people put alpha I/0, what they mean is that the alpha subunit is either one of the alpha I's, and there are 3 different alpha I's. There's alpha I1, alpha I2, alpha I3, or it's alpha 0. Okay, so when I put alpha I/0, that means a heterotrimeric G protein with an alpha subunit that is either one of the alpha I's or it's the alpha 0. And these all do pretty much the same thing. When they are in the on state, they go off and inhibit adenylyl cyclase enzymes. Okay, so let's say initially that it's in the off state here, and here is the beta-gamma complex bound to the alpha subunit in the off state. Okay, so I'll colour in the beta subunit in blue here, and the gamma subunit in green. Okay, right. Basically, if you want to actually produce a large enough amount of G-beta gamma subunits to go and activate phospholipase C beta enzymes in to a significant level, you actually need to activate GI/naught heterotrimeric G proteins. Okay, now why do you need to activate the GI/naught cascade? Well, the reality is that in cells, you have far more heterotrimeric G proteins of the GI/naught type than you do GQ/11. So basically, these types of heterotrimeric G proteins, these are really rare compared to these types. You have absolutely loads of these. Okay, so when you activate the GQ/11 cascade, you produce alpha Q/11 subunits in the on state, and you produce beta gamma complexes, but you produce very few of each. Okay, now the alpha Q/11 is extremely potent at activating phospholipase C beta enzymes, and therefore it doesn't matter that you only have a little bit of that. Okay, but the beta gamma complex is much less potent at activating phospholipase C betas, and the amount that you get produced of that isn't sufficient to significantly activate the phospholipase C betas. So instead, what you need to do is activate the GI/naught cascade by activating G protein coupled receptors that are GI/naught coupled. Okay, and a good example of a GI/naught coupled G protein coupled receptor would be the mu opioid receptor. Okay, so if you activate one of these GI/naught coupled G protein coupled receptors, you're going to get much more alpha I/naught being produced and much more beta gamma complexes being produced, simply because the cell has a huge number of this type of heterotrimeric G protein. So you get much more of the alpha I/naught than you would alpha Q/11, and you get loads of the beta gamma complexes. And then this huge production of beta gamma complexes that you get through the activation of the GI/naught cascade, this is now actually sufficient to significantly activate phospholipase C beta enzymes, specifically mainly phospholipase C beta 2, but also phospholipase C beta 1 and phospholipase C beta 3. Okay, right, so now how does the beta gamma complex activate phospholipase C beta enzymes? Well, it's pretty much the same as the way the alpha Q/11 activates the phospholipase C beta enzymes, except that it binds to different portions. Okay, so alpha Q/11 bounds to the already membrane-bound phospholipase C beta enzyme here, and it bounds to the coil core domain and the H-alpha-1, H-alpha-2 domain and reoriented the phospholipase C beta enzyme, specifically the catalytic domain of the phospholipase C beta enzyme, up to the underside of the phospholipid bilayer so that then the XY linker is repelled out. G-beta gamma is going to do pretty much the same thing, but it's going to bind to slightly different regions. So I'll just put the beta gamma here. Okay, so here is our beta in blue, okay, and here's our gamma in green. Okay, so where does the G-beta gamma complex bind? It binds to the plextrin homology domain here, which remember didn't really have a function in binding to phospholipids in these phospholipase C beta enzymes, and it also binds to the Tim barrel domain itself. Okay, so these are the two sites that the G-beta gamma subunit binds to, and again, what it serves to do is merely to reorient the Tim barrel domain up so that it's at adjunct to the underside of the phospholipid bilayer. That then repels the XY linker out, and then it can begin its process of breaking down PIP2 into diacylglycerol and IP3. Okay, right, so that's how the G-beta gamma subunit can activate phospholipase C enzymes of the beta type. Now, the one last thing that I want to mention is the synergism that you can get between alpha Q/11 and G-beta gamma subunits. Okay, so basically, you'll note that alpha Q/11 binds to a very different portion of the phospholipase C beta enzyme to the G beta gamma subunit, or complex rather. Okay, so in actual fact, phospholipase C beta enzymes can get both of these binding at the same time, and the activation that they produce together is much bigger than you would expect merely from activating the two together and adding on their effects. So let me explain what synergism means. Okay, I'm just going to go over the page where I've got more space to do this. So basically, let's draw a little graph for this. So on the y-axis, let's have phospholipase C beta activation level. Okay, and now here we'll have how much activation we get for a certain amount of alpha Q/11 that we have in the on state. Okay, and here we'll have the amount of activation we get for a certain amount of the beta gamma complex. Now basically, this might be the level of activation that we get for the alpha Q/11, this might be the level that we get for the beta gamma complex. We might expect that if we put both of them together, that you would merely get the two added on top of each other. Okay, you would merely get this line here added with this line here, so you would get a bigger line, but it would just be an additive addition basically. Synergism is the phenomenon that if you give them both together, so here's the alpha Q/11 with beta gamma, the actual effect that you get is much bigger than you would expect just from adding the two together. Okay, so that obviously means that when both alpha Q/11 and beta gamma bind to a phospholipase C beta enzyme, that the level of activation that they produce, the ability of them to activate that phospholipase C beta enzyme, is now bigger than what you would expect just from having the effect of the beta gamma added onto the alpha Q/11 subunit. Okay, so when both of them bind at once, it's really kick-starting this phospholipase C beta enzyme to actually start hydrolyzing PI45P2 into diacylglycerol IP3. Okay, so this is the origin of why, if you activate the GQ/11 cascade with the GI/0 cascade together, you can get a massive amount of PI45P2 hydrolysis, and therefore large calcium signals and large activation of protein kinase C, basically. Okay, so you can get much more hydrolysis than you would expect merely from the addition of these two pathways together. They synergize, basically, and you get a massive activation if you activate both of them together. Okay, right. So that now concludes our discussion of phospholipase C beta enzymes. Let's now turn our attention onto the next family that I want to discuss, which is the phospholipase C gamma enzymes. Okay, right. So firstly, let me tell you once again about the expression profiles for the two different phospholipase C gamma enzymes. So remember, there were two phospholipase C gamma enzymes. There was phospholipase C gamma 1 and there was phospholipase C gamma 2. Okay, phospholipase C gamma 1 is the one that's expressed in most tissues. This has a really broad expression profile. You find it in loads of different tissues. Phospholipase C gamma 2, meanwhile, like phospholipase C beta 2, is mainly found in the hematopoietic cells. Okay, so cells of the blood. Right, okay, so let's now talk about the structure of the two phospholipase C enzymes that are gamma type enzymes, because it does slightly differ from the structure that we saw for the phospholipase C beta enzymes. Specifically, it doesn't have that special H-alpha-1, H-alpha-2 domain, or the coiled domain. Okay, and this means that unlike the phospholipase C beta enzymes, which were capable of binding to the underside of the cell membrane themselves, the phospholipase C gamma enzymes are not capable of binding to the underside of the cell membrane themselves, and therefore, in their activation, you require two things. You require their recruitment from the cytoplasm to the underside of the cell membrane, and then you require the activation, the removal of the XY linker. And in the case of phospholipase C gamma, the removal of the XY linker is going to be done by different means than merely reorienting the protein so that the electrostatic repulsion can get the XY linker out. Okay, so it's going to be done by different means, and we'll see those different means in a moment. Okay, right. So let's start off then with the structure of the phospholipase C gamma enzymes. Okay, and this applies for both gamma 1 and gamma 2. So the first portion is exactly the same. You have your plextrin homology domain here, the pH domain. Again, it's not going to be at all important in these phospholipase C gamma enzymes, so it doesn't bind to phospholipids in these ones. And again, you have these two dimers of EF hands. Okay, so overall 4 EF hand domains. Okay, and again, they're not important here, but we'll colour them in nevertheless. So here's our plextrin homology domain, the pH domain, in orange here. Here are 4 EF hand domains, here in red, 1, 2, 3, 4. Okay, and then following that, what you're going to have is the catalytic domain, the tin barrel-like domain, which I'll put here. Okay, consisting again of the X box and the Y box. Okay, folded into this barrel structure. Again, here is the catalytic site at the centre of the tin-like barrel domain. Okay, right. Now, in these phospholipase C gamma enzymes, you're going to have a hugely magnified XY linker. So the XY linker in these ones actually contains loads of really interesting stuff, basically. Okay, so here is the portion coming off the X box here. And here is the Y box here, and this is the portion that's going into the Y box. Now, you have what's called a split plextrin homology domain. Okay, now what this means is that the two portions that are going to make the plextrin homology domain are split in the primary structure of the protein. Okay, but when the protein folds up into its three-dimensional structure, they're brought back together to make the full plextrin homology domain. So this is one full plextrin homology domain, but the two portions that make it up are separated in the primary structure of the protein. So this is what we would call a split plextrin homology domain. So this is our split pH domain. Okay, right. Then what you have is each of these continues on. Okay, and I'll draw this going around like this. So here is the continuation of the X box going on here. There's part of the split plextrin homology domain which is dimerized with this other part of the split plextrin homology domain, although they're separate in the primary structure. Okay, so we'll continue on the X box portion at the moment. Okay, so after this first portion of the split plextrin homology domain, you then have what's known as the N-terminal SH2 domain. Okay, so this stands for the N-terminal, that's the little N portion there. Okay, just meaning that it's the SH2 domain that's closest to the amino terminus of the protein. And then SH2 stands for Sarc homology 2 domain. Okay, and basically again, like plextrin homology domains, Sarc homology 2 domains, or SH2 domains, are domains that are found in a huge number of different proteins. Okay, but it was first found in Sarc family kinase enzymes, which is why it's called Sarc homology, and it's the second Sarc homology domain. Okay, right, so we're going to have another SH2 domain, and the significance of SH2 domains is that they're capable of binding to phosphotyrosine residues, so tyrosine residues that have had a phosphate group stuck onto them. So Y is the single letter code for tyrosine. Okay, so when you stick a phosphate group onto a tyrosine residue, that's then called a phosphotyrosine residue. And Sarc homology 2 domains are capable of binding to phosphotyrosine residues. Okay, and this will become important in a moment. Oops. Okay, there we go. Right, so there's the N-terminal Sarc homology 2 domain. Now we're going to have another Sarc homology 2 domain, which is going to be curved back like so, and it's going to be sitting in this catalytic site here. It's going to be occluding the catalytic site. So this is another SH2 domain here, sitting over the catalytic site, and therefore providing the auto-inhibition for the enzyme. Okay, and this SH2 domain is then called the C-terminal SH2 domain, or the CSH2 domain. Okay, so these are the two SH2 domains that these phospholipase C gamma enzymes are going to have. This one is closer to the N-terminus than this one, which is why this one's called the N-terminus SH2 domain. This one is closer to the C-terminus of the protein, and therefore it's called the C-terminal SH2 domain. But both of them are capable of binding to phosphotyrosine residues. That's the important thing to remember. Okay, then after the C-terminal SH2 domain, you're then going to have your final notable domain here, which is an SH3 domain, a Sark homology-free domain. Okay, and these usually are involved in binding to proline-rich regions of proteins. Okay, so portions of protein where you have a lot of proline. Now, for our discussion, we're not going to talk about the SH2 domain really at all. Okay, we're going to be concentrating on these two in tandem SH2 domains. Okay, so just to reiterate this then, here is the N-terminal of the protein. You go into the plextrin homology domain, the EF hand domains, then you have the X-box region here, then you leave the X-box region, have the first portion of this split plextrin homology domain, then the N-terminal SH2 domain, then the C-terminal SH2 domain, then the SH3 domain, then the other half of the split plextrin homology domain, then the Y-box, and now we need to do what's after the Y-box. And after the Y-box, all you have is then a C2 domain, okay, and then nothing else. Okay, you don't have that H-alpha-1, H-alpha-2 domain, or the coil-coil domain that we had in the phospholipase C beta enzymes. Okay, but again, this C2 domain doesn't play that big a role in the activation of the phospholipase C gamma enzymes. Okay, so therefore you have a much bigger XY linker. All of this mess that I've drawn here, all of this is the XY linker now, and again it's performing the same function of being auto-inhibitory for the phospholipase C gamma enzymes. That CSH2 domain is blocking the catalytic site and therefore is keeping the enzyme inhibited. Now, again, I'm going to repeat something that I said earlier, which is that because the phospholipase C gamma enzymes do not have that coil-coil domain that the phospholipase C beta enzymes have, they are not capable of binding themselves to the underside of the phospholipid bilayer. Okay, so these are always in the cytoplasm until they're actually going to be activated. Okay, so they're not in an equilibrium between being membrane bound and being in the cytoplasm like the phospholipase C beta. Okay, so this is cytoplasmic. Right, okay, so let's now discuss the principal ways that phospholipase C gamma enzymes can be activated. Now, the principal way that phospholipase C gamma enzymes can be activated is by receptor tyrosine kinases. Okay, so let me now talk a little bit about receptor tyrosine kinases, or RTKs. Okay, right. So RTKs stands for receptor tyrosine kinases, and basically these are a large family of receptors, okay, which are all characterized by usually having a single membrane-spanning alpha helix, and then on their intracellular aspect they have a kinase domain inbuilt to them. Hence why they are called receptor tyrosine kinases, because really they're a tyrosine kinase enzyme that then is also a receptor. Okay, hence why they're called receptor tyrosine kinases. Okay, right, so let me just show you the structure of a receptor tyrosine kinase. And if you want the sort of archetypal example to be thinking of, the archetypal receptor tyrosine kinase is the epidermal growth factor receptor, the EGF receptor. Okay, so the ligand for this receptor is EGF, which stands for epidermal growth factor. Now, a lot of the ligands for different receptor tyrosine kinases are growth factors, which are peptide molecules. Okay, right, so let me show you the structure of a general receptor tyrosine kinase, but the one that you can be thinking of in the back of your mind if you want a specific example to be thinking of is epidermal growth factor receptor. Okay, right, so the N-terminal of the receptor tyrosine kinase is extracellularly, so this top portion is extracellular, this bottom portion is cytoplasmic. Okay, and you'll have the ligand binding domain extracellularly. You'll then have the membrane spanning alpha helix. Now, I'm not showing it actually as an alpha helix just to make it look simpler, but this would be a membrane spanning alpha helix. Okay, and then on the intracellular side, you have a really important domain, which is the tyrosine kinase domain here, which is the domain that's capable of phosphorylating tyrosine residues. Okay, right. So this is the structure of a receptor tyrosine kinase. Okay, right. So what can now happen is when the ligand binds to the receptor tyrosine kinase, and we'll think of it as epidermal growth factor receptor. Okay, when the ligand binds to the receptor tyrosine kinase, like so, it will cause changing the conformation of the extracellular domain of the receptor tyrosine kinase. Okay, and this will now allow this receptor tyrosine kinase to dimerize with another receptor tyrosine kinase that has also got the ligand molecule bound. Okay, so here is the ligand molecule. Okay, so now what can happen is this can dimerize with another receptor tyrosine kinase, which I'll put here, okay, which will also have a ligand molecule bound to it. So I'll draw the ligand molecule here. Okay, right, and this is purely done for interactions with in the extracellular domains, basically. Okay, now, for the basis of this discussion, we'll keep this simple and we'll say that the dimer that is formed here is a homodimer. Okay, so the two receptor tyrosine kinases that have dimerized together are exactly the same receptor tyrosine kinases. So maybe the ligands here are epidermal growth factor, and these two receptors are the epidermal growth factor receptor, and they've dimerized together. In reality, the rules are a little bit more slack than that, okay, and you can form heterodimers, but to keep the discussion simple, we'll just imagine that it's a homodimer. Okay, right. Now, what then happens is you bring these two tyrosine kinase domains of these two receptor tyrosine kinases very close together. Now, the tyrosine kinase domain of a receptor tyrosine kinase, it's not completely inhibited. Okay, so it does have a vasoactivity. Its vasoactivity is just extremely slow. Okay, so before activation of the receptor tyrosine kinase domains, you have a very, very slow vasoactivity, so it can phosphorylate tyrosines, it just does it very, very slowly. Now, how can you activate the tyrosine kinase domains of these receptor tyrosine kinases? Well, each tyrosine kinase domain has a special tyrosine itself, which, if phosphorylated, will activate the tyrosine kinase domain. Okay, so if this tyrosine kinase domain gets its little tyrosine here phosphorylated, it will now become hugely activated, and the same for this tyrosine kinase domain here. Okay, so what dimerization serves to do is it brings the two tyrosine kinase domains really close together, and now what ends up happening is, because they have vasoactivity, eventually what will occur is this one will now phosphorylate this one's tyrosine phosphorylation site here. Okay, so it helps to number the receptor tyrosine kinase up. Okay, so let's call that one number 1, let's call this one number 2. So the kinase domain of receptor tyrosine kinase number 1 will phosphorylate this tyrosine phosphorylation site on receptor tyrosine kinase domain number 2. Okay, so here is that phosphate group. And now what of course will happen is that the kinase domain of receptor tyrosine kinase number 2 will reciprocate, and it will phosphorylate the tyrosine on tyrosine kinase domain of receptor tyrosine kinase number 1. Okay, right. And when they get their tyrosines phosphorylated like this, this causes them to become activated. Okay, so they're now going to have much greater activity. Okay, so they're now fantastic at phosphorylating tyrosine residues. And they end up phosphorylating a whole load of tyrosines on each other's intracellular aspects. Okay, so each one of these intracellular domains of each of the receptor tyrosine kinases has a huge number of tyrosine residues, and basically now the tyrosine kinase domains have been activated like so. What can happen is they can phosphorylate the tyrosines on the opposite receptor tyrosine kinases intracellular domain. So for instance, the tyrosine kinase domain of receptor tyrosine kinase number 1 can phosphorylate the tyrosine residues on the intracellular domain of receptor tyrosine kinase number 2 here. Okay, and then of course the tyrosine kinase domain of receptor tyrosine kinase number 2 can phosphorylate the tyrosine residues on the intracellular domain of receptor tyrosine kinase number 1, like so. Okay, and now this dimer of these two receptor tyrosine kinases with these phosphorylated tyrosine residues, which we've discussed, is called a phosphotyrosine residue. So we've now got loads of these phosphotyrosine residues. This serves as a starting point for loads of intracellular signaling cascades. So loads of proteins can come and assemble onto these phosphotyrosine residues on the intracellular aspect of the receptor tyrosine kinase dimer. Okay, now there's one key piece of terminology that I should put here, okay, which is that this process of getting all of these tyrosine residues phosphorylated is known as autophosphorylation. Okay, so auto means self, so it's self-phosphorylation, and you might think, well, it's not self-phosphorylation, it's the opposite receptor tyrosine kinase phosphorylating your tyrosine residues, so it's hardly self-phosphorylation. But if you view the receptor tyrosine kinase dimer as being one entity in itself, okay, then it is autophosphorylation. The tyrosine residues are getting phosphorylated by means of tyrosine kinase domains located in the dimer. Okay, right, so that's the origin of it being called autophosphorylation. So now what's going to happen is loads of different signaling proteins are going to assemble onto these phosphotyrosine residues. The one we're going to be interested in is the phospholipase C gamma enzymes. Okay, so both phospholipase C gamma 1 and phospholipase C gamma 2 can be activated by receptor tyrosine kinases that are activated in these dimers like so. Okay, so basically what's going to happen is the N-SH2 domain here of the phospholipase C gamma enzyme combines to phosphotyrosine residues on the intracellular aspect of the receptor tyrosine kinase. Okay, so how am I going to draw this? I would quite like to just bring this down here. The problem is I haven't got space on that side. I think I'll draw the receptor tyrosine kinase dimer again, so just bear with me for this. Okay, so here is the cell membrane. Okay, here is our receptor tyrosine kinase, at least one of them here. Okay, like so. Here's its C-terminus. Then we've got the other member of the receptor tyrosine kinase dimer. Here's the amino terminus. Here's the tyrosine kinase domain. Here's the C-terminus. Okay, and then they have both bound their ligand molecules, which we might assume is epidermal growth factor here. So here's one ligand molecule. Here's another ligand molecule. Okay, then they've activated each other by phosphorylating these special tyrosine residues, which are a phosphorylation site on the tyrosine kinase domains themselves. Okay, and after that what they've done is phosphorylated loads of tyrosine residues on the intracellular aspects of these receptor tyrosine kinase proteins. Okay, so here are these tyrosine residues, and now they're all going to end up being phosphorylated, and therefore we're going to have this appearance of loads of phosphotyrosine residues on the intracellular aspect of the receptor tyrosine kinase dimer. Okay, and now what's going to happen is proteins with SH2 domains are going to be able to come and bind to these phosphotyrosine residues, and this includes our phospholipase C gamma enzyme. So let me now show you the phospholipase C gamma enzyme bound to one of these phosphotyrosine residues. Okay, so I think I'll start by showing you the NSH2 domain. So I'll draw the tin barrel-like domain here. Okay, and it will have to be a bit smaller this time. So here's our tin barrel-like domain. Okay, split into the X box and the Y box regions. Then you have your split plextrin homology domain, which is here. Okay, now what I'm going to do is put that N-terminal SH2 domain being bound here, okay, to that phosphotyrosine residue. So it's the N-terminal SH2 domain which initially binds the phospholipase C gamma enzyme to the receptor tyrosine kinase. Okay, then you'll have the SH3 domain, which I'll put up here, okay, like so. And then at present, the C-terminal SH2 domain will still be covering the catalytic site, like so. Okay, so if I colour this all in, so we'll have the split plextrin homology domain here in orange, like so. Okay, we'll have that N-terminal SH2 domain in blue here, and it's the one that at present is bound to the phosphotyrosine residues of the receptor tyrosine kinase dimer. At present, the C-terminal SH2 domain has not bound to a phosphotyrosine residue. Okay, so this is the N-terminal SH2 domain. Here is the C-terminal SH2 domain. Okay, we'll then colour in the SH3 domain in red here. Okay, and then we'll put on the final other domain. So we've got that C2 domain, which isn't really doing anything. Okay, and then the C terminus, and then prior to the X box, we've then got the 4 EF hand domains here, 1, 2, 3, 4, and then we've got the plextrin homology domain, which is at the N terminal, which again isn't really doing anything, and here is the amino terminus. Okay, so just colouring in those domains. Here's the plextrin homology domain in orange. Okay, the 4 EF hand domains in red, like so, and the C-terminal domain, which we'll have in blue. Right, excellent. So basically, this first initial binding event, which binds the NSH2 domain to a phosphotyrosine residue on the receptor tyrosine kinase dimer, all this is doing is bringing the phospholipase C gamma enzyme from the cytoplasm to the underside of the cell membrane. So this is the relocation step. This is the moving the phospholipase C gamma enzyme from floating around in the cytoplasm to being underneath the cell membrane. Okay, now what we need to do is the activation step, which involves getting this C-terminal SH2 domain out of that catalytic site of the tin barrel-like domain there. Okay, so we've now got it up ready underneath the cell membrane, out of the cytoplasm, which was something we had to do, remember, because the phospholipase C gamma didn't have that coil-coil domain that the phospholipase C beta enzymes had, okay, and therefore couldn't attach to the cell membrane on its own. Okay, so now how are we actually going to get the C-terminal SH2 domain out of the catalytic site? Well, this involves the tyrosine kinase domains of the receptor tyrosine kinase dimer now phosphorylating tyrosines on the XY linker, okay, and elsewhere on the phospholipase C gamma. However, there is one tyrosine that is believed to be utterly important in this, okay, and it's this tyrosine that I'm going to give you the name of, and this tyrosine is between the C-terminal SH2 domain and the SH3 domain, okay, and this tyrosine is called tyrosine 783. Okay, so that just tells, oops, not tyrosine 787, tyrosine 783. Okay, so that's just telling you what position it's in, and the 783rd residue turns out to be in between the C-terminal SH2 domain and the SH3 domain here. Okay, and basically what's going to happen is one of these tyrosine kinase domains here in the receptor tyrosine kinase dimer is going to end up adding a phosphate group onto that tyrosine residue at position 783. Okay, and turning it therefore into a phosphotyrosine residue. And then what happens now is the C-terminal SH2 domain here ends up binding to that phosphotyrosine residue in the phospholipase C gamma enzyme itself. Okay, so this ends up binding to this phosphotyrosine residue in the XY linker. Okay, this changes the conformation of the XY linker and ends up removing the C-terminal SH2 domain from this catalytic site, and that is then how you open up that catalytic site, and then it's available to actually now allow a PIP2 molecule to stick its head in and hydrolyze that PIP2 molecule to diacylglycerol and IP3 and produce the signaling pathways associated with that. Okay, right, so that's how you activate phospholipase C gamma enzymes using receptor tyrosine kinases, and you'll note that this mechanism of getting the XY linker out of the catalytic site is very different from that reorientation mechanism that relies on the electrostatic repulsion between the XY linker and the underside of the phospholipid bilayer. This involves the phosphorylation and then a conformational change in the XY linker which removes it from the catalytic site. Okay, right, so we'll call it there for this video. In the next video what we'll do is just finish off our discussion of phospholipase C gamma enzymes by discussing the fact that phospholipase C gamma 2 can also be regulated by the monomeric G protein RAC2, and then we'll go on to the phospholipase C delta enzymes.